Hello, I'm Dr. Pamela Ruig, and I hold the David J. Ellis Chair in Large Animal Clinical Sciences at Michigan State University. And in today's video, I'm going to be discussing contaminated milk samples. Now, all of us who've submitted milk samples for mastitis cases to a lab for culture have sometimes gotten the result that the sample's contaminated. And it's super frustrating, because we get these results back, we go to all the work to collect the sample, and then we can't use the results. So in today's video, I'm just going to spend a few minutes to talk about what contamination is, how it happens, why it matters, and how to prevent it. Now, to understand contaminated milk samples, we have to think about what causes mastitis. And mastitis occurs most of the time when a single bacterial pathogen breaches the teat canal, gets into the udder, and causes an infection. So store that away. Most of the time, mastitis occurs when a single bacteria causes an infection. So when we recover more than one bacteria from a milk sample of a cow with mastitis, we've got to try to figure out which one causes the infection, or is it a contaminant? Now, the National Mastitis Council has definitions for how to interpret milk samples that are cultured. And they define the occurrence of two bacteria that grow from a milk sample to be called a mixed sample. But when there are 3 or more different colony types visible on the plate that we're culturing, that's called a contaminated milk sample. So 3 or more different colonies growing on a plate from a single milk sample is the standard definition of contamination. Now, that sounds really straightforward, but we use different types of augers in different labs, so sometimes it's a little more complicated to interpret what contaminated is. In a conventional microbiology laboratory, we usually take the milk samples into the lab and we'll put them on a standard media, blood agar, and it grows everything. And so we'll often put maybe 1 milk sample on each half of a blood agar plate, and then we'll look at that plate after incubation 24 hours later and see what's grown. On that type of plate, it's really easy to see if there's different colony types. One colony may be small, one colony may be big, they may have different colors, they look visually different. And if there's 3 different ones growing from that milk sample, it's easy to call that contaminated. Now, the difficulty is in many of our on-farm laboratories, we use selective augers. So we may have 2, 3, or 4 different selective medias on a single plate that we put that milk sample on. Each of those selective augers only grows certain types of bacteria. So you won't see different colonies on each of the augers. So in selective medias, if we see growth on the gram-negative and the gram-positive and the strep, and there's different colony types there, we know it's contaminated even though we don't see different colonies on each segment. So that's an important thing to recognize. The contaminant plates may appear different depending on the agar that you've got. Now, how do samples get contaminated? Now remember, intramammary infections occur when bacteria go through the teat canal and infect the quarter. We take the milk samples from the quarters to diagnose the pathogen. And the most common reason we get contaminated milk samples is errors in sampling. So I would encourage you to review our video that shows and describes aseptic collection of milk samples, because that's the most common reason we get contamination. There are some other reasons. Occasionally, samples are mishandled. In other words, they're not cooled fast enough after collection. Or sometimes in the laboratory, someone might sneeze on a plate and contaminate it. But number 1, recheck your sampling procedures. Then we get to, well, we've got these contaminated samples back. Can we use the results? The answer is no. If we have a sample that's got 3 or more colonies in it, we really have to go back and resample the quarter. And that's because while mastitis is caused by bacteria, there's also a lot of other bacteria in the cow's environment and on the teat skin that can easily get into that milk sample. E. coli are in fecal matter everywhere. Non-aureus staph are on teat skin and in the street canal. And if those teat ends aren't fully cleaned with alcohol, it's easy to get contamination. So we do have to resample those that are contaminated. Now let's recap. 3+ colonies growing from a single quarter milk sample indicates that sample is contaminated. The results from that contaminated sample can't be used. We have to go back and redo the sampling. And remember, the single most important thing we can do to prevent contamination is to follow the proper procedures for aseptic sampling.